בבקשה, פרופסור וילנר, הבמה שלך, אפשר להתחיל.
יופי. Good afternoon to our Chinese participant. Good morning to the Israeli participant. I, Professor Itamar Villner, member of the Israel Academy of Science, and Professor Yuliang Zhao, member of the Chinese Academy of Sciences, act as co-chairs of this bilateral meeting on nanotechnology. The Israel Academy of Sciences has long-term relationships with the Chinese Academy of Sciences. We praise the advances in science in China, and the relationships and cooperations between the two academies are reflected by many scientific meetings in various fields, exchange of scientists, students, and most importantly, friendships. Israel would like to advance and promote the activities in China and to help the advance of scientific activities in both countries. 2 years ago, we decided to host in Jerusalem a bioacademic meeting in the area of nanotechnology. Unfortunately, due to the corona pandemic and several delays to conduct a frontal meeting, we decided to realize this effort by having a series of 4 webinars in several subtopics in the area of nanobiotechnology. The 4 webinars addressed the following topics: nano devices and nanophotonics, energy and environment, self-assembly and biomaterials, and today's the 4th and last webinar on nanomedicine and nanobiology. I realize that many scientists from both countries are disappointed for not being able to present their research in these webinars, yet I hope that you will understand that due to the time differences between the countries and the time restrictions and limitations, we were able to organize only the meetings with the active participations of only a few scientists. While preparing this meeting, we had to overcome many obstacles in organizing this webinar series. And I would like to thank my co-chair, Professor Zhao, and his team from the Chinese Academy of Science for the continuous help and support. I would like also to thank the scientific chairpersons, moderators, and speakers that participated in all these webinars and are doing their job also today during the last webinar. Particularly, I would like to thank the staff of the Israel Academy of Sciences, Mr. Galia Finzi, the director of the Academy, Joe Cohen, the director of International Relations, Naama Shiloni, head of Public Relations, Yaakov Rotman, who is responsible on the computing at the Academy, for their help to organize this series of seminars. I would like also to mention Mr. Arik Futterman, the technical producer of this meeting that you met today during our preparation. Last but not least, I would like to thank Dr. Yael Ben-Chaim, the Secretary of the Science Division of the Israel Academy of Sciences. Her daily efforts to organize this series of webinars and her sleepless nights and efforts to organize this series has contributed to the success of these events. On behalf of Professor Neely Cohen, the president of the Israel Academy of Sciences and Humanities, I am delighted to welcome all the participants that will participate in today's meeting. As the co-chair, Professor Yuliang Zhao was unable to join me for some opening remarks due to other commitments. I'm honored to invite Professor Zhizhang Wei from the National Center of Nanoscience and Technology to greet the participants. I wish all of you a successful and stimulating meeting. Please, Professor Wei.
Thank you, Professor Itamar Wiener, and good morning and good afternoon, respectful Professor Itamar Wiener and ladies and gentlemen. I'm Zhixiang Wei, Professor and Deputy Director of National Center for Nanoscience and Technology. Welcome to the China-Israel Bilateral series in nanotechnology. We have successfully organized 3 sessions in early stage. We are very grateful for the efforts for organizing committee. Personally, I took part in the 2nd workshop as a speaker and the 3rd workshop as a chairperson. Today, I will give a welcome remark on behalf of Professor Yuliang Zhao, the co-chairman, and also the Chinese Academy of Sciences. China and Israel have made great achievements in the collaboration of nanoscience and technology area. The platform construction have encouraged continuously and friendly exchange of ideas and peoples. The collaboration has displayed fruitful highlights in talent cultivation, bilateral meeting, and joint project collaboration. The Israel-China bilateral series in nanotechnology mainly focuses on the latest progress and future development trends of nanoscience and technology. It provides a high-level exchange platform for scientists in the field of nanoscience and technology, and laid a solid foundation for long-term exchange and collaboration in this field. As we can see from the previous meetings, China and Israel have lots of the common interest in the area of nanotechnology, including nanodevices and nanophotonics, energy and environment, self-assembly, and biomaterial. And I'm sure we will find more common interest in nanomedicine and nanobiology today. I hope this such collaboration will provide an outstanding opportunity for academic exchange, technology innovation, discussion on the future collaboration, exchange of the people, and share our research prospects as well as resources. Finally, I need to thank Dr. Yu Xiao for the organization from NCST for this collaboration, and thanks for all the organizers for the success of the bilateral conference. Finally, I wish a great success for today's conference. Thank you.
Okay. Good morning and good afternoon, everybody. My name is Yael Khanin. I'm Professor of Electrical Engineering from Tel Aviv University. And it's a great pleasure for me to chair this session. So this session is about nanomedicine and nanosystems. And in particular, the effort today will be to focus on bioapplications, including imaging, fighting microbial infections, cancer therapy, and targeting tumor. And we have 4 lectures, as in the previous sessions in this conference, 2 from the Israeli side and 2 from the Chinese side. What I would like to remind the audience is please make sure to send your comments and questions so we can use them for addressing these specific issues later with the speakers and during the panel when we conclude this session. And so to get started, it's my very, very great pleasure to introduce Professor Alfa Benny from the Hebrew University. She's Associate Professor at the School of Pharmacy. She has her BSc degree and Master and PhD from in biotechnology from the Technion, did her postdoc at Harvard, and established her research at the Hebrew University, studying therapies and for drug delivery, but also focusing on new engineering-based applications and methods, how to better study the interface between the delivery and the biological systems. So, Ofra, it's your stage.
Thank you very much. I would like to thank, of course, Professor Wilner and the very distinguished moderators and chairman. Thank you for the great opportunity. So indeed, I'm coming from the School of Pharmacy, which is located at the Faculty of Medicine. The Faculty of Medicine is actually close by to our hospital. The campus in Enkerem is a campus that combines both basic research and medical or clinic practice. My lab is located in this campus, and we are working on three main topics. One of them is the tumor microenvironment. The other is bioengineering, and I include here microfluidics, 3D printing, very advanced 3D models of cancer cells, and of course, nanomedicine and drug delivery. So as you all know, there is a very major paradigm shift in the way we treat cancer these days. It's no longer one size fits all. We're trying to tailor therapy per individual, per person. The genomics and all the big data allows us now to profile the patients much better than we used to. And actually, there is a very major trend in moving into more targeted therapies. And when I'm saying targeted, it means both in the active compound level, meaning that the drug itself can be more selective, but also, of course, in the aspect of drug delivery. So we can target the location, the site, the kinetics much better. So we all know here, I guess, in this audience, the concept of the EPR effect, the enhanced permeability and retention effect. So we know that we can now actually utilize this very specific physiology of cancers, and we use the deformable or the defective blood vessels in order to design such drug carriers or nanoparticles that they can leak out of abnormal vessels in the tumor site, and they can remain more in this site and not in normal tissues. Now we can, of course, make it more advanced if we decorate nanoparticles using all kinds of ligands that allow affinity, enhance the retention, and of course, enhance selectivity. So this is a well-known concept, and it's being used for many years now in nanomedicine and cancer. But there is a major limitation when we are thinking about targeting or active targeting, and we cannot ignore this problem in drug delivery in cancer. And the problem is that cancer cells are not very stable genetically. We know that. We know they are constantly changing, so the surface molecules are not constant. So basically, if we're trying to develop and design very specific affinity moiety for a surface marker, we cannot be sure that this marker will stay for the whole course of disease or during the treatment even. So for some years in my lab, we were trying to think of another property that maybe will be a little bit more stable, maybe more universal in terms of how to target cancer cells. And we came up with the maybe unconventional approach of targeting, which actually utilized the biomechanics, the biophysics properties of cancer cells. And what do I mean by that? So we know from our basic knowledge in cancer biology that cancer cells has this tendency to move around. They are more deformable, they softer, they know how to adjust to different biophysical environments. And if we are looking at the literature, we can see that indeed, if we measure elasticity of cancer cells compared to normal cells at the same tissue, we see that cancer cells are more elastic, they are more deformable, and they are less stiff than normal cells. And we asked ourselves, maybe this also reflecting on the way that cancer cells can actually take up carriers, drug carriers. So the first experiments we've done was to take particles that are completely inert. These are polystyrene particles, let's say plastic particles per se. And this red color fluorescence show how cancer cells are very effectively can take up relatively large particles without really the particles has any function. We compared the uptake between more metastatic cancer cells than less metastatic ones, and we found that the more metastatic the cells were, the more efficient the phagocytosis of the uptake were. In this movie, you can appreciate how this uptake occurs and how extensive the particle uptake can be. So basically, the whole body of the cancer cells can be filled up with inert beads. So we know for years that there are many mechanisms of particle endocytosis and uptake. So if we discuss very small particles, let's say below 100 nanometer, we will probably have more dominant active endocytosis using clathrin and caveolin. But when we go towards more submicron or larger beads, we're actually now discussing more deformable mediated uptake, which is similar to phagocytosis in immune cells. And this was intriguing for us because then what we asked ourselves, maybe this biomechanical or the different biomechanicals of cancer cells can indeed affect the capacity of particle uptake. So we know that in order that uptake will occur in cells, we have an interplay between adhesion and cell deformation, and there is a sweet window in which a certain particle will internalize into a given cell if there is sufficient adherence and if there is enough ability to deform. We can, of course, model all the energetic consideration physically, but the point is that maybe based on that, we can find this window that only cancer cells can have sufficient deformability in order to engulf these particles that are difficult to engulf. That was our hypothesis for the mechanical targeting of cancer cells. So one of the key experiments that we've done was to even determine whether phagocytosis capacity of cancer cells is an innate property. And this is, I think, a very, very interesting study because here what we've done was to take a certain cancer cell population and we actually generated two subpopulations out of it. One that were more phagocyte-like, so they uptake a lot of particles, and the other subpopulation was such cells that did not uptake at all. And we sorted the cells, we generated it after a couple of cycles, and then we had stable two subpopulations, phagocytosis and non-phagocyte cancer cells, and we did it for melanoma and for bladder cancer cells. And then we start to compare these cells that came from the same origin. And what we found is that actually the more phagocytic cells were more elastic, as we predicted, but interestingly, the whole genome profile and the epigenetic profile was also different, and the more phagocytic the cancer cells were, the more aggressive they were. And this is very interesting because it means that just primarily by sorting cells by phagocytosis, we can say something intelligent about their aggressiveness. So that means that maybe we can use it also as a diagnostic principle. So we actually summarized a lot of data, and we were able to validate this triangular correlation between cancer malignancy, their capacity to uptake particles, and their deformability or biophysical properties that are different than normal cells. So this is one aspect of more the biological concept, but eventually to study this, we need more systematic tools to control particles and nanoparticles. And we know that we can control size and shape, composition, particle stiffness, and surface texture. But these all needs tools, and we need tools to control them in a very precise way. So my lab works for many years now to develop microfluidic-based technologies in order to fine-tune properties of nanoparticles. So we showed that we are able to produce nanoparticles and microparticles at the same device, flow device, focus flow. We actually published it, and it's a very powerful tool for us because, as you can see, the control over the size is very precise. Now we can control other properties as well, shape, if it's gold nanoparticles, minerals, or we can also control porosity. And each of these properties have implication of how it interacts with cells. And the final step that I want to mention is that eventually we want to take all this knowledge and provide tools for personalized medicine in cancer. And for that, we want to be able to grow cancer cells in a more physiological mimicking way. And for that, we actually developed a clinical protocol that we can take biopsies from cancer patients, we can disintegrate the tissues, and then actually we build their tumors once again, but now instead of one, we can do it for hundreds of microtissues, and we can do it in three-dimensional way. So basically we have panel of microtumors from the same patient, and now if we want, we can actually screen different cells or screen different drug vehicles to try and tune the right therapy per person. So we're doing organ-on-chip technology. We actually develop way to 3D print microfluidic chips, as you can see here, with very good resolution and good transparency. So we can now do this Lego-like modular designs for tissue on chip, cancer on chip. And we can 3D, to grow them in 3D and to visualize them in high resolution. In this slide, it's just a taste of one of the clinical validation data that we have. This patient carried brain cancer, glioblastoma. She was a young woman with very advanced brain cancer. In order to do a compassion study, we were trying to grow her tumor in our tumor-on-chip technology. And interestingly enough, we were able to detect a very rare mutation that is more abundant in, is more common in breast cancer and not in brain cancer. And actually this mutation has a certain drug that can target it. We tried to treat the patient. Unfortunately, it was too late for her, but this gave us the idea that we can actually detect mutations that we cannot do otherwise because of the depth of sequencing. So just to summarize, I showed you that biomechanical properties is important. We can use it as a targeting approach. My vision and my lab vision is that mechanomics will be part of this scheme that will be given to any cancer patient, and it can be for diagnostic and for precision therapy. I'd like to thank my group, especially Dr. Ifat Bril-Karnieli, who is a biophysicist that I showed many of her data in this presentation, Bentzi Amoyev and Eliana Steinberg that are working on the microfluidics, and all the other students, and of course the funding agencies that supporting our research. So thank you.
Okay, thank you very much, Alfa, for a wonderful talk. I think Shlomit can start with the questions.
Yes, I see. I don't see any question in the question and answer, but what I would like to ask is, I have a question about the microfluidic system and the spheroids. If you put the spheroid in the microfluidic system, like what size of channels you have and how exactly you analyze what's happened to the spheroids?
Yeah, so if we do it by 3D printing, of course, the resolution is a little bit more limiting than fabrication, like photolithography. But what we can do now is actually 27 micron is the minimal resolution that we can, or the maximal resolution that we can get in the XY, and 1 micron in the Z. So it's quite good for organ-on-chip. The spheroids themselves are around 200 micron. And what we can detect is, of course, all what we can by imaging, which is viability or all kind of markers. We can stain them for immunostaining and so on. But of course, we can also connect sensors. And that's something we're trying to do now for real-time detection of nutrient and oxygen consumption.
And do you get also vasculature in the spheroid?
We didn't try to do vessels on chip, although there are chips like that and we can do that. What we did found is that if we co-culture endothelial cells along with cancer cells, they actually form a kind of network. It's not functional, but we do get the cues and the cross talk between the cells.
Thank you.
I would like to ask another question.
Sure.
So you showed very nicely that you can engineer different particles and control pretty much many different properties, including, most importantly, the mechanical features. How much do you expect the mechanical properties that you determine is going to be affected in vivo, so, for example, by protein attachment and so forth?
So that's a key question, because so far, of course, all the concept that I just showed here is more at the cell level, because the interaction is between particle and cell, exposed cell. Of course, in vivo, we have the extracellular matrix, we have protein kind of binding and so on. I don't have a clear answer yet. I think we have to choose the indication in a smart way. So it probably will not be a very established tumors, but more early stage tumors and so on. So the cells are more exposed. But this is something we're doing now, and hopefully in the coming year, I will have more, smart answers for that.
Professor Ni has a question.
Oh, yes. Yeah. Thank you for pointing me to ask a question. So Professor Benny, very interesting talk. So I think the mechanical targeting is really a cool idea for targeting tumors. So my question is for your uptake fingerprint. So I just want to know, have you seen from different stages of the cancers, for example, from beginning and to more malignant, and then to long distance metastasis, have you seen quite different signatures for this group of tumor cells for one type of tumor, for example? And the second one, I'm not quite sure I understood. You say malignancy and deformability. I see certainly there is a link. So how about the uptake capacity? So is this uptake for nanoparticles or other like micro objects, or is uptake about, for example, other glucose, other nutrients, or is there a specific properties or in general uptake for everything? Yeah, thank you.
Yeah, so thank you. So I'll answer the two questions. The first was about the site of the malignancy level, and we do see that there are different patterns of uptake between different sources or origins of cancer cells. If they are more malignant, they actually uptake differently. And the second question is very important, actually, because right now we focused on inert particles for a reason. I mean, we didn't want any affinity. So right now the whole concept is in inert and only biomechanical driven uptake. But we are sure that if we will look at other components like glucose or any other affinity, possibly affinity groups, it will change the pattern. I believe the pattern of the uptake would still be different, but I guess there will be a shift. So once we select the particle, the chosen particles, and that can be barcoded, it can be more than one, we want to stick with them. Otherwise, it changes the old patterns. So at least for diagnostic, we are about to do so. I mean, we are going to select a certain particle mix, and then we should stick with that. Otherwise, it changes the data.
Thank you. Are there any other questions from the panelists? Thank you very much, Alfa.
Thanks. Bye-bye. And thanks for all the organization. Thank you.
Okay, so I think Professor Wu can continue to introduce the next speaker.
Okay. The next speaker is Professor Guangjun Nie. Professor Nie received his PhD degree from Institute of Biophysicals, CAS. In 2008, he joined the National Center for Nanoscience and Technology. Dr. Nie was awarded the National Distinguished Young Scientist Award in 2013. Professor Nie is a chief scientist of the National Basic Research Programs from 2012 to 2016 and 2018 to 2022. Dr. Nie is serving as an associate editor of Nanoletters. Professor Nie is working in the field of functional nanobiomaterials for regulation of tumor microenvironment and intelligent anti-tumor nanorobots. Dr. Nie has authored over 200 peer-reviewed papers, which have received a total citation of 12,000 times based on Google Scholar, comprising papers published in Nature Biotechnology, Nature Biomedical Engineering, and Nature Communications. Dr. Nie is the founding chief member of Materials Biology and Intelligent Diagnostics Technology Division, Chinese Biophysical Society of China, serving active scientists in the multidisciplinary field of biology, biotechnology, biomedical engineering, and material sciences. Today, Professor Nie will show us his topic is the biomolecular-based nanostructures for tumor microenvironment, targeting and regulation. Let's welcome to Professor Nie.
All right. Thank you, Igor, for your very kind introduction. Before I start, I really want to acknowledge the Chinese and Israelis colleagues to organize this webinars. Although this is the fourth one, but I think it's still very attractive. I see a lot of people very interested in joining this webinar. So today I will talk about our recent work based on biomolecules nanostructures for their tumor targeting and regulation. So I share a very common interest with Professor Alfa Bani. So we're also interested about the tumor micro environment, especially the blood vessels. So for the because my background is in biochemistry, so I'm familiar with the biomolecules. So here we work with all kinds of biomolecules, from peptides to nucleic acids, and also biomimetic hybrid structures. We use them to modify their cell assembly properties and to make different nanostructures. So hopefully we can use such new structures to regulate tumors. So I always saw this the image for tumor microenvironment. So we know there are many players in this TME. So for example, today I will talk about more like vasculature regulation, especially endocellular cells and platelets. They are the major players in the vasculature TME. For sure, there are more other targets for design responsive and accessible nanostructures. I think we have several strategies for tumor vasculature regulation, from the inhibition of angiogenesis to induction of tumor coagulation. And on the other hand, we can also increase the drug acceptability by various means. For example, enhancing the EPR effects by specific depletion of tumor-associated platelets. So altogether, so basically either we can block the blood flow or to increase drug acceptability. So based on different tumor types. So the first case I show you is we work with the specific peptide. This peptide is part of the degradation byproducts of a protein called integrin. And this peptide here we call it 616Y. It already been shown has the capacity to inhibit the tumor angiogenesis. But the problem is that the peptide degrades very fast. People cannot achieve therapeutic purpose. So in this case, we combine a hydrophobic domain with the hydrophobic peptide, make a amphiphilic structure. And specifically, we integrate a protein, a molecule called the DAP. So basically, it's a pH sensor, has a pKb value around 6.8. So with such structure, we can use it to regulate, for example, the endocellular cells migration properties. We measure the phosphorylation properties of FAK, PI3K, and AKT, all of the key players downstream of integrin pathways. We found that the DAP616Y works much more potent than the free peptides. And then we look for the tumor cells. We look for their, for example, first is degradation of galectin, and the second one we see their invadopedia formation. So all of the properties associated with the migration of tumor cells, we found that DAP16Y peptide works much more stronger than the free peptide too. And when we work in the in vivo model, here we use a xenograft tumor model. We test either the free peptide and DAP16Y and also a crumbled control peptide. So in this case, we found that the 616Y combined with the DAP form nanostructure inhibit more potent for tumor growth and also decrease the tumor macrovessels density. So I show you one case how we work with the tumor blood vessels, and then because we know tumors have enriched vessels and also very high density of blood vessels. So we think if we can either slow down the blood flow or totally block the blood flow, probably can achieve more potent anti-tumor activities. So in this case, we designed a DNA-based nanostructure. In this structure, we made a rectangle DNA structure and mobilized 4 molecules of protease. They can trigger the blood coagulation. And more importantly, we modified the nano rectangle nano sheet with the so-called targeting strand and also recombinant. The targeting strand can find the recombinant nucleolin, which you can open up. So in this case, we make a nanostructure that can be open and closed, depends on the local environment. So we call such DNA structure a DNA nanorobots because there are thrombins on top of the nano sheets. So this closed open property can specifically induce tumor blood coagulation. So basically, we're turning impossible vision into reality. So here we show some cellular studies. In this case, we found that the DNA nanorobots, the closed open status depends on binding of endocellular cells, which have overexpressed nucleolin proteins on the cell plasma. And this is in vivo study. When we inject the DNA nanorobots into the tailwind of the mice, we see a distribution in the first few hours, and then start accumulated the fluorescence signal of the DNA nanorobots and reach the plateau around 8 hours, and then degraded along with time. And after 24 hours, the fluorescent labeling nanorobots almost disappeared. And we see that the accumulation turn into induction of the tumor local coagulation. And then we try different tumor models, start from the MDA, MB 231 human breast tumor models. We try different control groups. We found that only the nanorobots with a targeting motif and also loading with thrombin can significantly inhibit tumor growth. And also more importantly, we found that the treatment group has a dark color. So it's a direct sign of the formation of thrombosis. And tumor bearing mice showed improved survival activity. And then we try other tumor models, for example, this B16F10 melanoma model, and also this is poorly permeable human ovarian SK-OV3 tumor model. All the models shows that only the DNA nanorobots can enhance mice survival. And finally, we try another model. This is a doxycycline-induced lung tumor model in the tight-on systems, which will induce a KRAS G12D mutation. In this case, if we give the mice in the water of doxycycline, the mice will start to express the KRAS mutation and induce local multi-module lung cancers in their lung. And the nanorobots can inhibit the tumor growth by this MRI image and also tumor staining. So after showing the therapeutic efficacy of various tumor models, we want to confirm such DNA nanorobots are safe. In this case, we use both mice and the mini pig as models. We measure their blood biochemistry indications and also the blood flow and also tissue staining. We found there were no severe side effects be observed, meaning no blood coagulation formed. Okay. So I saw all of the study in this as a cartoon. So in this case, we mimic the liver blood supply, and you see the small DNA nanorobots circulating in the blood. It's closed stage, only open up when they reach the protein on the surface of tumors and open up the structure and expose their loading protease and activated platelets and form the blood coagulation. And this one is very specific, only happened in the tumor tissues, not in normal tissues. Okay, the paper published gets some attention from the field. In this case, optimal leading targeting of thrombin to tumor coagulation. Following this previous study in last year, we did another further study. In this case, we found that because we know that whenever we treat the tumors, although multiple tumor types respond for blood coagulation therapy, but after a certain time, when you stop the treatment, the tumor grows back. So when we look closer for the tumor tissue, we found that although there is a big tumor necrosis core, but on the surface, there is always a tumor rim. They are alive cells. So when you stop treatment, they will grow back. So that's probably tell us what's the problem, why the tumor grow back. And the tumor burning mice, they got dead eventually. So in this case, we combine the blood coagulation with the chemotherapy. In this case, we made a nanostructure, this nanosphere with the thrombin and also the chemo drug dogs. So in this case, we can use 2 punches to hit tumors, and both the blood coagulation and chemotherapy work more effectively to induce the tumor cell death. So in this case, we combine 2 therapeutic strategies can work much better than one. So I show you a couple of cases. We work with the inhibition of tumor blood supply. On the other hand, sometimes we want the tumor vasculature is more permeable. For example, we want more like antibody-based therapeutics, and even cells, T cells can get into the tumor tissue. So in this case, we found that tumor platelets, they stick to the tumor vessels and make the tumor vasculatures more uniform. So we want to specifically deplete tumor-associated platelets. How we can do that? In this case, first we invest what are the specific biochemical environments of tumors. We found that MMPs, the family, normally overexpressed, activated in most of cases. So in this case, we choose MMP2 as a responsive protease. They can cut their substrates. And for the nanostructures, we used a MMP2 substrates coating the surface, and then inside we combined the platelets binding specific antibodies and also the chemo drugs. So in this case, we can achieve tumor specific release of the platelets binding antibodies. And when the antibodies bind to the platelets, they got a microaggregation, and such aggregation will be phagocytosed by macrophages. So in this case, we can achieve our purpose. And so I show you some of our key data. So this is the nanospheres, and they can co-load antibodies and chemo drugs. And when the tumors has a high expression of MMP2s, the fluorescent labeled nanostructures accumulated more in the tumors. See this compared data. And we also use other imaging modalities to show indeed the for example, both the photoacoustic imaging and PET imaging can show that this is MMP2 responsive coding can lead to up to 3-fold increase in tumor accumulation of the drugs. And here we show you some tissue level staining. Here we found that indeed the tumor vasculature get damaged and more blood components released from the blood vessels. And we found that both tumor types, for example, the MCF7 and A549, these 2 types of tumors, they express different levels of MMP2s. Both of them show enhanced increased activities of the chemo drugs. So finally, I just want to sum up what I proposed today. So we work with tumor vasculatures. We can use different nanostructures, can target to TME. We can make a TME responsive. We can also remodel the TMEs based on different purpose. So finally, I want to sum up my study. So what I want to propose here is based on the only those people, the traditional therapeutics can work out. But now if you have make the right medicine by drug design and based on the early diagnosis to give to the right people using the targeted delivery can give the drug to the right place and control the drug release at the right time. I think they probably represent one of the futures of anti-tumor drugs. Thank you very much. Thank you. That's all for my talk.
Thank you, Professor Nies, for your nice talk about the tumor microenvironment targeting strategies. So, any questions?
I don't know how much time I spent.
It's fine.
Actually, there is time for definitely there's time for questions.
Yeah. So whenever I focus on the screen, I can't see what time left. So I probably speak a little faster than normal.
About 20 minutes, 18.
Okay.
So anyone raising your hand? No, maybe I have a question for you. How about the platelets, the targeting strategies for the compared to the non-conducting? How about the targeting rate? I mean the ratio between compared to without the targeting. We use the platelet strategies.
Okay. Thank you, Aigo. I think Aigo asked me the question. So what first is what's the difference with the platelet targeting and not? And probably, and you have another meaning is that what are other methods to target, like for example, increase the blood permeability, right? So first is by using some imaging methods, we can confirm that the accumulated drugs, by all means, we can increase couple folds, for example, 3 folds by the PET imaging. So meaning, so if you can deplete the platelets beforehand, you can have a 3-time increase about the drug accumulation in the tumor. And
okay,
there are many other methods. For example, using natural oxide, you can also dilate blood vessels. Probably you can also increase the drug accessibility. That's from literature, not from our own study.
Okay, thank you. And there is a question from Professor Alfa Benny. His question is there a potential development of immune responses against the DNA carriers?
Sorry, can you say again, because I've looked for another one. So
can you see the chat room?
Yeah.
Chat room, in your chat room.
Oh, okay.
Yeah, Professor
Okay. Okay. Thank you, Alfred. This is a cool question. I think from the study, the people know that when the pathogen invade our systems, they always release some nucleic acids, and some of them are like really immunogenic. But so far, well, I can say that one is sequence dependent, the other is structure it's a sequence dependent, the other is a length dependent. So meaning so we have the way to tune because our structure is based on single short strands of DNA, so we can test them beforehand. And in our study, although we haven't carried on very in-depth immune study, but from our current understanding, there are no severe immune response for the immune system. But there are maybe ways to further fine-tune the structures, for sure. Yeah.
Thank you.
There is another question.
Yeah, there's another one. How do you estimate the right dose needed for effective cognition? Okay.
Yes.
This is also a very important critical question. So people won't want to get overdosed, for sure. And for us, we targeted to for example, the mini vasculatures of the tumors, and so we need quite a large amount of the thrombin. For example, we load 4 molecules into 1 nano robots, and now we also work with 6 or 8 and compare what are the optimal doses for that case. So far, we think it's okay if we load for 4 or 8 molecules per nanostructure, because we found that when we inject the first time and when we look for the tumor tissue, we found some of the tumor tissues get reopened. For example, they have an anti-system against the coagulation. Okay. But relatively is not that easy to do the in vivo titration for the optimal doses. But for so far, we try different molecules per nanostructure. They also similar trends. Thank you.
Okay, thank you, Professor Nie.
Okay, so thank you very much. And we move on to the next speaker. So it's my pleasure to introduce Professor Udi Benin. He's professor at the Faculty of Life Sciences at Bar-Ilan University. He also received his bachelor degree in biotechnology from the Technion and his PhD from Tel Aviv University, then followed up with a postdoc position sorry, at the University of Washington in Seattle, at the University of Iowa. And his research focuses on microbiome films and the understanding those films and also targeting them as an engineering approach. So Udi, it's your stage.
Thank you, Yael, and again, thank for the organizing committee for this wonderful opportunity and sharing with you what we're doing in the lab. And I'm going to take you a little bit away from cancer in the next 20 to 25 minutes, and I hope you'll find it as an interesting story. And what I'll try to do is start with a short introduction about the challenges that we're now seeing related to infectious diseases, and tell you about 2 projects in the lab that we've been developing to try and tackle these challenges in a novel way. So I think the last year and a half has put forward the issue of infectious diseases. So not saying that it wasn't in the front, but really the coronavirus pandemic put forward how susceptible mankind is to infectious diseases. And I think the fear against future virus pandemics, as well as the growing understanding from our side that we're seeing more and more antibiotic resistant as a challenge, is 2 of the major problems that us, the microbiologists, and of course other researchers dealing with antimicrobial development are now facing. And looking at the past, I think probably one of the biggest discoveries in terms of medicine was the discovery of penicillin by Alexander Fleming over 90 years ago. And this really changed how humankind lives and changed medicine, how we do medicine. And really following this discovery, there were, I call it the golden era of antibiotic discovery. For about 20 to 40 years, microbiologists and chemists around the world really biochemists discovered most of the antibiotics or the building blocks of the antibiotics that we know today. And it was really a golden era it was such a hype around it that even the Surgeon General of the United States somewhere around in the mid-60s said we can close the chapter on infectious disease and then focus on, I would say, not new diseases, but diseases that were starting to have their impact, like cancer that we've been talking in the last few lectures. And unfortunately, many drug companies started to lose interest from different reasons to look for novel drugs. And we see a decrease in novel antibiotics that come into the market in the last 20 to 30 years. And that is a huge problem because if you look at the bacteria and the microorganisms, they have not stopped working on finding solutions to these antimicrobial agents. And this is really a fascinating figure. I don't know how many of you are aware of it, but on the right side, you have when the antibiotic was introduced into the community. And on the left side, you have when a resistance was formed. So for penicillin, even before we introduced it into market, we already knew that there is resistance. But following that, a few years, 2 to 5, 10 years, following introduction of an antibiotic, we start seeing resistance. And this cat-mouse game is something that we are dealing it for since we introduced penicillin, like 70 years, a little bit less than that. And in the last 15 to 20 years, we're hearing more and more concern from health agencies around the globe that we're hitting the bottom of the well in terms of novel antibiotics, and we're slowly entering to what is called the post-antibiotic era, going back to the times before Fleming discovered antibiotic. The last report came a few months before the corona pandemic, I would say the most serious report telling us, listen, the scientific community, the industrial therapeutic community, we have no time to wait. Governments around the world, we really need a solution. Some of the solution is, of course, developing new antimicrobial agents, but they propose additional solutions that will minimize the transfer and evolution of antibiotic resistance. And I apologize to my Chinese colleagues, but I'll briefly say what's written here in Hebrew. This is basically a piece from a newspaper article saying that the antibiotic resistance is going to be the corona pandemic to look like a child's game, because the numbers are frightening, and it is speculated that in about 20 to 30 years from now, most of the deaths, the #1 death cause will be from antibiotic-resistant bacteria. It will surpass cancer. Okay, and the impact, and we've seen what infectious diseases can do to the world and do to our living and do to our ability to give solutions quickly to the community. And of course, the economic impact is going to be really substantial on governments. So we really need to find novel solutions in terms of fighting infection and antibiotic resistance. Now, my lab has been working on this subject for the past 15 years. And one of the things we've focused on is one method by which bacteria actually enhance their resistance to antibiotic. And this is by diverging into a specialized mode of growth called biofilm. So you all know that bacteria live as free-living single-cell organisms, and that is called the planktonic state of growth. But bacteria can actually switch into a biofilm mode of growth, where they form a structured community in which they're enclosed in a self-produced polymeric matrix. And this mode of growth immediately increases the resistance to challenges and to hostile environments. And this could be disinfection, exposure to antibiotic, and the immune system. So basically, even a susceptible microorganism, a bacteria, once it goes into a biofilm, it goes into this shelter and it is protected from the antibiotic and can survive the treatment. And I don't know how many of you are aware, but we're all infected by biofilms. This is the plaque that we have on our teeth and on our gums, but biofilms are all around us. And this increased resistance to stress and to a chemical challenge and our immune challenge has caused biofilm to be a major problem of contamination, both in the environment and in the industry, the water industry, the food industry, and of course, the focus of our session, medical. So most of the chronic and hard to treat bacterial infections, so people here who suffer from chronic urinary tract infection, chronic sinus infection, etc., you have biofilms in your body, and this is why it's chronic, and this is why the antibiotic treatment and your immune system is not able to get rid of it. Many of the infections are associated with medical implants, catheters, as I'll show you in a minute, heart valves, orthopedic additives that we add to our body. These abiotic surfaces are perfect for colonization of the bacteria. Now, how is biofilm formed? So I don't know how many you know or familiar with the 3 or 5 second law or rule, which states that if food falls on the floor, you have about 3 to 5 seconds to pick it up before it gets contaminated. So this is not true. Actually, it gets contaminated in milliseconds, so bacteria will attach very quickly. It is true that the initial attachment is reversible and many will detach from the surface, but given enough time, this reversible attachment becomes irreversible, and bacteria will start growing on the surface, secreting this matrix that holds them together. And in a few hours, you will start getting microcolonies of bacteria, giving, depending on the conditions, additional hours and days, you will get the mature biofilm. We all know it from what's happening on our teeth if we don't brush our teeth well every morning and every evening. And the last step is, of course, active or inactive dispersal of the biofilm, where free living organisms are detaching and looking for new places to colonize. Now, my lab have been working very extensively in the last 10 to 15 years, together with chemists and material scientists and engineers, on developing novel anti-biofilm strategies. And nanotechnology, and this is why I chose the title from microbiology to nanotechnology, has been a very important tool in our work, because using nanotechnology, we can actually either enhance or actually get new antimicrobial activity, and the ability to nanofabricate and really better assemble antimicrobial agents and other approaches on the surface can really expand our toolkit, our much needed toolkit, to fight this bacterial colonization and infections. And what I want to try to do do today with you is tell you 2 stories of our approach on fighting the first step or blocking the first step of biofilm formation, and this is adhesion. And the idea is that if the bacteria cannot adhere in the first place and form the biofilm, the initial stages of the biofilm, then we can probably minimize the contamination and minimize the infection altogether. And I'll show you 2 approaches. One is microbe repelling, and one is based on biocide release. So basically implementing antimicrobial agents on the surface in order to protect it from colonization. But I want to start with the microbe repelling, because I think this is really, I would say, an elegant and very interesting approach in which you don't have any antimicrobial activity on the surface. Rather, what you do is you nanofabricate or change the surface in such a way that it will repel the microbe. So it's like the microbe gets closer to the surface, there's a no entry zone, not because it is killed, but actually because it's not convenient for it to settle down. And people have been working on this approach quite extensively, changing the wettability. So some of you may have heard about superhydrophobic surfaces, changing the charge or the topography of the surface. And I want to tell you about an approach that we've been using together with a startup company that utilizes surface acoustic waves. So you're all familiar with ultrasound, which we use extensively for imaging, but there's also additional surface waves called Rayleigh waves or surface acoustic waves that actually do not penetrate the tissue as much, but vibrate the surface. And a startup company at the northern part of Israel called Nanovibronics has developed a device called the UroShield, which they apply to catheters. So this is a catheter, this goes into your body, This part is outside, and you hook the uroshield on the external part, and this generates surface acoustic waves on the catheter, both on the inside and the outside. And we were able to show that once you apply surface acoustic waves on the catheter, and remember, there's no chemistry here in terms of releasing materials, nothing whatsoever, just the vibration. You actually, once this methodology applied, you need a much higher inoculum of bacteria in your urine in order to get biofilm form and an infection. Okay, so the ability of the bacteria to grow in urine is difficult to begin with, and once you need more bacteria there, you minimize the ability of the infection. And even once biofilm is formed, the biofilm is much more susceptible to antibiotic activity if you apply the antibiotic in the presence of the surface acoustic waves. So you can actually even treat better once a biofilm is formed using this device. It was also shown that this can actually delay catheter-associated urinary tract infection in an animal model, a rabbit catheter animal model. And this was very reassuring. But really, the Holy Grail came from a clinical trial in Europe in which 27 patients were tested. 70% of the control patients that were walking with a catheter without the uroshield got infected. All of them, by the way, by a single type of bacterium called Pseudomonas aeruginosa, which we extensively work in the lab, one of the most problematic bacteria now in hospitals in terms of multi-drug resistance. Only 20% of the patients wearing the uroshield got infected. By the way, none with this notorious microorganism Pseudomonas aeruginosa. And this is really reassuring, and now this product is actually can be purchased. It has the past regulation in Europe, and I think some time ago also in the States. And I like this project because this is one of the projects that actually started very applicable in my lab, okay, very applicable research to try and see whether this technology can inhibit biofilm formation, but became actually a basic project that we now study because this made us realize that bacteria have a bioacoustic response. So of course, bacteria don't have ears, they don't hear sound, but they can respond to it. And once we started looking at the bioacoustic response of Pseudomonas aeruginosa, we saw that actually in addition to minimizing biofilm formation and increasing its susceptibility to antibiotics, there's also reduction in virulence. So even if the bacteria do stick around and form some biofilm, it is much less virulent to the host than the one that is formed without surface acoustic waves. And we were actually very successful in starting to understand this bioacoustic response, and we identified a bioacoustic response regulator. So this is a transcription regulator that somehow responds to the surface acoustic waves. We're still looking for the sensor, but once this is activated, we get these phenotypes, okay? Reduced expression of virulence factors, it reduces biofilm formation, and the biofilm that is formed is much more virulent. So this SOR that we called it, the specialized bioacoustic regulator, it could be a very interesting drug target, even for in terms of finding chemical agents that might influence its activity. So this is the first approach of actually making the surface uncomfortable, I would say, for bacterial colonization. The other approach, which is much more widely used, and you can actually find products like this, is what I call the Biocide Release. And here the idea is that you impregnate the surface with an antimicrobial agents, and this could be antibiotics or biocides or antimicrobial nanoparticles, as I'll show you in a minute, anti-quorum sensing, etc. And these materials slowly are released, and of course, incoming bacteria to the surface are hitting this before they attach to the surface. This kills them or affects their activity, and no biofilm is formed. And this market of antimicrobial coatings is really growing up fast, and it is expected to double from 2019 to 2026 with over $6 billion in terms of the market. And I think the COVID-19 just put this growth even to a faster accelerance. And I'm sure most of you remember that in the first months of the pandemic, people were very concerned how long the virus can live on surfaces. So if somebody releases aerosols of the virus on a surface and somebody comes after a few minutes and touches it, what will happen? Will it get infected or not? So the idea of having an active surface that can kill pathogenic microorganisms, bacteria or viruses is very important these days, and I'm sure you can all appreciate it. And I'm going to tell you two stories how we challenge this dogma. And the first one is really a great collaboration with Professor Aaron Gedanken from our university, from the chemistry department. And Aaron really brought into, developed very extensively the approach of sonochemistry. So I don't know how many of you are aware about sonochemistry, but the idea is that you use the ultrasound and the energy released by the ultrasound and the sonocavitation to both synthesize the nanoparticle, but also using the microjets from the sonocavitation also coat surfaces. So it's in a two-in-one process. Both the synthesis of the nanoparticles and the coating is done under one treatment. And we've been using this technique quite extensively with many, many papers focusing on making antimicrobial metal oxides on nanoparticles and putting them on the surface, antibiotics, and even enzymes. So you can actually retain the enzymatic activity. And in the last few years, we actually started playing with what I call doping of metal oxides. So instead of having pure metal oxides like zinc oxide or copper oxide, we started to try and making combinations of them. So actually having zinc and copper together at different ratios and making a doped nanoparticle. We were very successful in doing zinc copper doped oxide nanoparticle. And what was great about this new agent was it was much more active against both sensitive bacteria and multi-drug resistant bacteria compared to the pure, I would call, zinc oxide or copper oxide nanoparticles. So it kills better at lower concentrations and much more faster. So this is a very active nanoparticle. And we actually use this novel compound to coat, again, catheters, which are a very serious cause of infection, nosocomial infection in hospitals. So you can see the coated catheter and the uncoated catheter, and we coat the catheter using this methodology, both from the outside and the inside. I can tell you today that there's a commercial system being made that is deposited in Ganya Silicon, a company in the northern part of Israel that is testing now this technology. And once we coat the surface, we can dramatically reduce biofilm formation on the surface of common urinary tract infection pathogens. And again, using the same model I reported for the surface acoustic waves, this rabbit model, we were able to show that we can dramatically reduce catheter-associated urinary tract infection in this animal model. So it's much, the infection and the colonization in the urine system is much lower when you have this coated catheter compared to the regular catheter we routinely use in hospitals. And the biocompatibility and biosafety of this agent, of this coating, was proven as well using various testing. So there was no apparent problem with the animals in terms of histopathological and hematology, no serious leaching from the catheter, etc., that can damage the host. So biosafety and biocompatibility is very high with these coatings. And a few years ago, a company actually took the technology, a company called Sonovia, mostly actually taking it towards textiles. And then the COVID pandemic came up, and suddenly a new market of masks came to awareness of the company and all of us. And they took this research one step forward and actually tested the coding on coronavirus and found this metal oxide coding to be very active. And they came up with a product called the Sonomask, which was actually very successful here in Israel. We're hopefully losing masks by next week in terms of the need to use them here in Israel. But the technology really, this showed the proof of concept of this technology, both in real scenarios and in real life activities. Okay, so the last Biocide Release story I want to tell you in my lab actually comes from a different problem we started to see, that there are, I would say, consumers in which the regular Biocide Release approach is not sufficient, because in many cases, after a few months or a year, once this antimicrobial agents is diffused out eventually at the end or diminishes, you have to throw away and buy a new product. And this is good maybe for masks or for catheters, but it is not relevant to products that people hope that will work for several years. So there's no regeneration ability in many of these coatings. In situ, I would say regeneration. And looking at this problem, together with another fruitful collaboration here at the BIU with Professor Shlomo Margel, also from the chemistry department, Shlomo and his students came up with a really interesting approach using N-alamines. And N-alamines are basically compounds that contain a nitrogen and a halogen atoms. And the idea is that the halogen is in a state where its oxidation state remains in such a way that it still has its biocidal properties. But the good and important factor of N-alamines is that once the halogen is depleted, the compound can actually be regenerated upon exposure to an active halogen such as sodium hypochlorite or bleach. And we saw that we can very effectively synthesize nanoparticles of these agents, and these agents are very active in terms of their antimicrobial activity. Furthermore, they actually have a very high stability compared to bleach, to I would say the golden standard of halogen treatment. So you can actually expose much more our compounds at the same concentration of active chloride in terms of the compound or the amount of, I would say, bacteria exposed to the agent. So while bleach exhausts itself very quickly, our compounds can withstand several cycles of exposure. And do I need to finish? This is the sign?
Yes.
24 minutes. But we're running a little bit late, forward. So if you give me 5 more, I will appreciate it, if it's okay. Right, I need to finish by 12:40, I think, right? 12:40. So it will take me 3 more minutes or 4 more minutes. So
go ahead.
I apologize, so it takes a bit too long. It's very active, but it's not as active as bleach. And I think this, I would show you in a minute, that this actually comes as a better approach, because we don't want to kill very quickly. We only want to kill the bacteria that stay on the surface. So bleach kills in terms of few seconds to minutes. This takes a little bit longer to get killing, but once it's settled down, the bacteria on the surface, it will get killed. I'm stuck for some reason. I apologize, but I'll stop sharing for a second because it got stuck. My apologies. Okay. Can you hear me? Because for me, on my end, it got stuck. So I don't know what the reason for it.
We hear you.
From your end that stops me, or because I went over time, but for some reason, it's not moving forward.
No, we hear you, Udi. You can go ahead.
Oh yeah, I know, but it doesn't move forward for some reason. So I don't know why that is. If it's something from my end or so, I'll try to fix it. I apologize. Can you see my screen, by the way, just to make sure?
Right now, you're not in screen sharing.
Screen share. So I'll start screen sharing again, and hopefully solve the issue. Okay, great. So for proof of concept, we actually combined our collaboration with Netafim. Netafim is a company that is leading in terms of water irrigation, and they invented or really improved the dripping system. For those of you who are not familiar, the idea is not to water the whole field, but you actually put water only by the root of the plant, so it drips, but they suffer from biofouling. And biofouling blocks the dripper, and eventually what will happen is that some of these will get blocked and the efficiency of the field will reduce. And I'm sure many of you are not aware, but actually to prevent this, what people who grow plants do is actually chlorinate the water or use biocides, either routinely, constantly, or in high concentrations every 2 or 3 weeks in order to stop biofilm from forming. So what we thought of offering Netafim was to actually put our nanoparticles, chlorinate them, and basically what will happen is that you run water, and especially this is an issue with when you reuse sewage water, which we do in Israel a lot for irrigation. Bacteria, only the bacteria who sit for about 20 to 30 minutes will get released and killed, so we get the biofilm cleaned up. And once it is exhausted from the dripper, all the farmer has to do is actually what he's been doing constantly anyway, run some chlorine in the system. This will get recharged, and we will get get the system up and running. And we were able to actually implement our nanoparticles in the drippers, and we can see that we can recharge them. And we went into the field, and this is real field study. And you can see that the regular drippers of Netafim, after a month without any chlorination, you see biofouling. Our drippers that came up with the chlorine on the dripper were clean. And this is visible on microscope, so you can see the fouling, no fouling on the treated samples, and limited organic carbon on the drippers that were chlorinated. And this can actually withstand for 5 months. So now the protocol means that you need to only chlorinate once a season. So you chlorinate in the beginning of the season, and that's it. You don't need to use any chlorination. So to summarize, I think what the main message I want to convey is that we really have no time to wait in terms of fighting infectious diseases, viruses, bacteria, etc. I think there's many challenges ahead of us, but we can discuss them later because I've been running over time. And again, apologize for this, but thanking the people, Professor Margel Gedanken, my students, Michal, who worked on the drippers and the nanoparticles, and Yossi, who worked on the surface acoustic waves, and of course the funding sources. And thank you very much.
Thank you very much, Udi. And Shlomit can now handle the questions.
Thank you very much. Are there any questions from the panelists or from the Q&A? Someone want to ask? This is the time.
If there's, I can ask one question is that you emphasize, Udi, several applications, and clearly this is a very, very applied and economically very important field. Can you comment a little bit generally about the economic viability of these solutions? Relatively speaking, I expect this field to have less regulatory constraints, but still probably very, very sensitive to cost. Can you comment about that?
I would start with the drippers, because there it's probably the most. I mean, a dripper costs a cent, and you cannot come with a $10 solution on top of it. But so this has been a challenge, and I'm happy to say that for the drippers, we reduced the price for making it attractive for netafim, for example. The rest of the solutions are also, I mean, surface acoustics is I mean, if you take into account how much the access of hospitalization, and if you look at the statistics, urinary tract infection, mostly due to catheters that is put in the hospital due to problems in health or problems after surgery, the cost that you put into it in terms of insurance companies, of course, governments, etc., is huge. So finding solutions that cost a few dollars on it is reasonable. Concerning regulation, yeah, that's an issue, and I think that's part of what drew away companies from looking into antibiotic discovery, because it still costs a lot to put a novel antibiotic on the market, but it's not a blockbuster drug anymore, because this will sit in a vault till someone will take it out and use it until, you know, which is reasonable. You don't want to use a new antibiotic. First, you'll use all the old ones, and only if there's resistant, you'll use it. So you really need to change the model of how you compensate companies for antibiotic development and really pushing them towards this activity. And I hope that what happened with COVID and the awareness now that how we can impact, how this impacts our life will change this strategy and really put money into where we really need it, because the situation is bad.
Thank you very much. If there are no more questions, thank you very much.
Thank you.
Okay. The next speaker is Professor Mingyuan Gao. Dr. Gao received his PhD degree in 1995 at Jilin University, China. He spent 6 years as postdoctoral research fellow and group leader in Max Planck Institute of Colloidals and Interface and the University of München, respectively. He became a full professor in the Institute of Chemistry, Chinese Academy of Sciences in 2001. He moved to Suzhou University in 2020. His current research interests include the creation of molecular imaging probes and their biomedical applications. Today, Professor Gao will show you his talk is Smart Probes for Cancer Imaging. Let's welcome Professor Gao.
Thank you, Agua. Thank you very much for your kind introduction. Hello, everyone. It's really my great pleasure to be part of this wonderful program. To be frankly, it's the first time for me to join the online meeting. But if I remember correctly, this is the second time for me to join this Israel-China bilateral series meeting. So I'm very happy to have this opportunity. And yes, over the past 20 years, I've been working in the Institute of Chemistry, Chinese Academy of Sciences. That is just one block away from the National Center for Nanoscience and Nanotechnology. And beginning of this year, I moved down to Suzhou University. Actually, I was invited to found a center called Molecular Imaging and Nanomedicine in 2013. And this center is officially opened in 2015. And just beginning last year, I started to establish my own group. And actually, it's very close to Shanghai. And if you travel from Hongqiao Airport to my office from point to point, you only need 60 minutes. So you're welcome to visit us. Then after the COVID-19 pandemic is brought under control. So today I will talk about a very classic biomaterial that is iron oxide, magnetic iron oxide nanoparticles. And I will share with you 3 stories. The first one is about the surface effects on the T1, T2, dual modality magnetic resonance imaging performance of small iron oxide nanoparticles. After that, I will talk about glutathione-induced particle aggregation for sensitive MR imaging that will be followed by quantitative mapping of glutathione in vivo through MRI. It's well known that our magnetic iron oxide particle hold great potential for biomedical applications. For example, an article published in Journal of Clinical Oncology summarizing clinical cancer advances 2015 spans a whole chapter to discuss about nanomedicine. And in this chapter, it specifically mentions iron oxide, saying that cancer-directed iron oxide particles have magnetic properties that make them suitable for use as imaging agents. They can precisely pinpoint the cancerous areas on MRI scans which surgeons rely on to plan for surgical removal of the tumor. Almost 25 years ago, FDA approved the first iron oxide particle-based contrast agent that is known as Ferdex. It is used for detecting cancer and lesions in liver because the normal liver tissue will uptake the particle and then produce a dark signal. And the same company, they invented Ferdex, also developed another iron oxide particle-based contrast agents a few years later. It's called Comidex. And after intravenous injection, part of the dose will travel to lymphatic system. And then it will be captured by the mononuclear phagocyte cells in the benign lymph nodes. In consequence, the benign lymph nodes will appear dark or even black. However, the metastatic lymph node will not uptake the particles, and then they appear light in color, which really gave a very great promise for detecting metastasis through magnetic resonance imaging. Unfortunately, sorry, here is blocked. These two drugs, the production of these two drugs has been ceased approximately 10 years ago, and it is said that the efficacy remains to be improved. Okay, and why it has unsatisfied efficacy? Then we got a Ferdex sample, and this image was captured in my lab. You see the particles appear very messy, and it's hardly identify individual particles. Yeah, simply because those particles are produced in aqueous system with a lot of kinetic parameters involved. In consequence, it's very difficult to control the particle size distribution, and also crystallinity degree. Fortunately, at the end of last century, and Paul Alexopoulos, I think most people know this guy by his famous quantum dots. He invented a new approach. They produce very uniform iron oxide particle by pyrolyzing a ferric organic compound in high temperature environment. And now in literature, you easily find protocols to tell you how to produce such uniform particles with different size and even different scales. For example, now people says you can easily get 100-gram scale of those beautiful particles. Nevertheless, those particles are hydrophobic because they are modified by in most cases, oleic acid. Then they cannot directly be used as a contrast agent. And taking the advantage of this non-aqueous synthetic approach, we spent more than 10 years to develop this method, and now we can we can directly get water-soluble and even biocompatible iron oxide particle out of this non-aqueous system. Yes, then talking about biomedical applications in organic nanoparticles, there are a lot of studies showing that the PK, PD strongly depend on particle size. And briefly, small particles, upon proper surface modification, they present longer blood half-time and shorter biological half-times. Biological half-times. That means small particles are preferred if we talk about in vivo applications. Now let's come back to iron oxide particle. Iron oxide particles are known as T2 contrast agent, but in recent years, people learned that when you reduce the particle size down to certain threshold, for example, one particle size below 5 nanometer, then they present strong T1 contrast effect. In the meantime, the T2 effect is suppressed. Then we ask whether we can enhance the T2 effect, in the meantime, gain strong T1 effect by reducing the particle size. And to answer this question, we prepared 3.6 nanometer hydrophobic nanoparticles. Then we also designed 3 PEG ligands. And the molecular weight of the PEG ligands are 2,000. Only difference is anchoring group, which brings surface ligand firmly on particle surface. The first one contains a disulfide group. The second one contains a hydroxamate group. And the last one is ketral group. Then we replaced oleic acid ligand. Then we get pegylated particle. We did very careful experiment to be sure the organic content in sample are identical or comparable. And moreover, they will not form particle aggregates in solution. And in this way, we can compare their relaxometric properties. And here the results tells us that the first one, the PEG bearing disulfide anchoring group, after it's coated the particle, the resulting particle present only T1 effect, not T2 effect. But the rest two, they give both strong T1 effect and T2 effect. Then we revisit the theory and come to a conclusion that due to the presence of this p-pi and pi-pi conjugate structure in the anchoring group, the inhomogeneity of the local magnetic field is increased by the ligand, which is also the origin of T2 effect. In consequence, we get particles, they show both T1 and T2 contrast enhancement effect. And for example, by using particle coated with this ligand, we can increase the contrast, T1 contrast by 33% and T2 contrast by 54% by the same particle. Yeah, this is very useful to exclude, to achieve precise diagnosis through MRI. Although we can increase the T2 effect by surface engineering, however, the effect is still limited. And from literature, we know the smaller the particle, the lower the T2 effect. But we also know that if we make particle aggregates, then the T2 effect will dramatically be increased. Then we asked whether we can cross-link the small particles within tumors to increase the MR imaging sensitivity. To answer this question, we designed such a particle probe. We designed such a ligand and to replace the hydrophobic oleic acid. In this ligand, we introduce a disulfide bond. You know, in most solid tumors, glutathione is upregulated, and it can react with the disulfide bond to generate a thiol group. And in this way, we hope the thiol group will react with monomer group from adjacent particle. In this way, it will glue the particle together. Moreover, we also introduce RGD peptide to increase the binding affinity of the resulting probe to tumor cells. And here we also introduce a peptide sequence. It's called self-peptide to increase the blood circulation behavior of the resulting particle. And to see the responsive behavior, we also design a control ligand in which the disulfide bond is missing. And we named particles modified by this ligand as non-responsive probe, and this one is responsive probe. Apparently, in the presence of glutathione, the non-responsive probe does not form particle aggregates. Here is hydrogen scattering results. In difference here, the responsive one, they form bigger and bigger aggregates. In the same time, the solution is becoming blurry. And in consequence of this particle aggregation, then the T2 effect is dramatically increased. And to show the effect of the self-peptide, we also did in vitro cell experiment, and the results suggest that the self-peptide modified particle, no matter it's responsive one or non-responsive one, the cellular uptake is much lower than the mother particle. And to further show the function of the self-peptide, we also labeled our particle with radioisotope in order to trace the particles. On the other hand, we can also use it for SPECT imaging. And the results shows that the self-peptide can effectively increase the blood residence time. Here's the blood half time from 2.8 to 6.4 and 8.2. Actually, this one is a little bit longer than this one, probably because the responsive probe gradually lose this self-peptide during the blood circulation. Now let's move to the final imaging results. In here is the non-responsive probe group. This is responsive group, and the quantified T2 value is given in this plot. If you compare the delta T2 for the responsive one with that for the non-responsive one, is remarkably increased. And then through a SPECT, we also can quantify the quantity of the probe in tumor area, and it says that the responsive one, compared with the non-responsive one, the signal from the responsive one is increased by 1.4. But if you compare the delta T2 from here to here with the non-responsive one, the signal is increased by a factor of more than 3, which strongly suggests that the particle aggregation forms within tumor. It's also worth to mention that if you look at this axle, you see the enhanced window is remarkably broadened up to tens of hours. Normally, for individual particle probe, it only stays within tumor for a few hours, then it will be washed out. But in this case, it stays much longer. And now I will share with you the last story. It's about the quantitative mapping of glutathione in vivo through MRI. And for this study, we slightly modified the previous responsive probe. We replaced the self-peptide with angiopeptide 2 in order to send the probe across blood-brain barrier for detecting intracranial tumor in the brain. And we still keep the RGD because we use it to detect tumor. And here shows you the characterization results. And in principle, in the presence of glutathione, again, it can form aggregates, and here as time goes on, the size of the aggregates keep increasing until 2 hours, then it's almost stopped. Okay, and then here, in comparison with the non-responsive one, you see the non-responsive, the black curve, they slightly fluctuate, but remain nearly unchanged. In comparison with that, then the responsive one gave time-dependent delta R1 and delta R2. For delta R1, it's decrease. For delta R2, it's increase. Then we, in order to increase the detecting sensitivity, we plot this two signal in the way that we use the ratio between delta R2 and delta R1. In this, we can increase the sensitivity for showing the responsiveness to GSH. Okay, and here you see again is very well inconsistent with the latter dynamic scattering study, and after approximately 2 hours, the signal reached the maximum. And if we incubate our probes in GSH of different concentrations, again, delta R1 will decrease and delta R2 will increase. Then finally, we get a similar curve, and it increased dramatically with the increase of GSH concentration, which covers the concentration range for interstitial and intracellular fluids of tumor. And in order to understand this correlation, then we write this chemical reaction equation here, and to through this process we get particle bearing thiol groups. This is the starting point for the particle to glue together. And here is the formation kinetics for this species. And then we, after a number of approximations, then we established a correlation between delta R1 and concentration of particle aggregates, and delta R2 with the particle aggregates concentration. And finally, we get this quantitative relationship between the ratio of delta R2 and delta R1 and GSH. Then using this equation, we fit this results, we get excellent data. Yes. And then we also did in vitro experiment to show the cell binding affinity of the responsive probe, and suggest that the responsive one really show remarkably increased cell binding affinity in comparison with the control probes. And here the responsive particle also present excellent accidentally low cytotoxicity. And finally, we use subcutaneous tumor as model. Then we detect the GSH in vivo. Then we got this plot. Then we feed with our theory. We get very good results here again. And finally, let me show you the final results on the intracranial tumor imaging. And here, this row are recorded from control probe. This two recorded from the responsive probe. You'll see in comparison with the control probe, our responsive probe presents much stronger signal than the control ones. And let's take this one, for example. The signal appears approximately 1 hour post-injection, and then reach maximum 3 hour post-injection. And as time goes on, the lower part of the tumor, the T1 signal is decreased. You see here, but the upper part persists for nearly 9 hours. And here, if you look carefully, it's just give a reverse tendency, which suggests has higher concentration of glutathione. This can better be seen if we extract the cross-sectional signal vertically. You see, 3 hours post-injection, this T1 signal, this is T2 signal. As time goes on, lower part present reduced T1 signal, but the T2 signal is increased. Then at 7 hours post-injection, this is completely reversed. Yes, this strongly suggests that the lower part of the tumor has a higher GSS concentration. To exclude other possibility, we also did the following study. For example, through staining, we can be sure that the particle distribution vertically are very even. Yes, and also through this immunohistochemical analysis, we can show that we found that the expression of glutathione rectase, yes, is higher in the lower part. And also this marker is for the proliferation of cancer tumors is also higher at the lower part. And with all this data, we finally can map the expression of GSH in this intracranial tumor. Here you see the skew bar is really, you can read out the concentration there. Okay, that's almost the end of my talk. I hope I have convinced you that the tumor microenvironment triggered responsiveness of smart probes I mentioned today may offer possibility for sensitive detection of tumors, and also the responsive property can also be used for quantitatively sensing the tumor abnormal signatures for disclosing their malignancy in vivo. Okay, so I would like to thank my colleagues and also collaborators and also the agencies for financial support. So I stop here. Thank you.
Thank you very much. And thank you very much for the talk. And we
it's
have
here,
yes, we have here a question in the Q&A. And
with those that containing component tumor reacted with another group, should we avoid this containing common tumor cells? This is a good question. You're talking about thiol containing compounds in tumor cells. I don't know in what kind of level and also what are they, to be frankly. But at least through our in vitro and in vivo study, we do see the glutathione induced responsiveness of this probe. If there is some thiol molecules in tumor cells, I don't think we can avoid its reaction with the particle, but it will absolutely decrease the sensitivity, that's for sure, because it will stop the aggregation process. Yeah.
Are there any other questions from the panelists? You're welcome to ask. Okay, so thank you very much. If there are no more questions,
yeah.
Thank you again.
Thank you. Thank you.
Okay, so thank you very much for all the speakers. And I think now we can turn to the panel discussion, which of course is open for questions from the audience, but otherwise we will probably start the discussion. Let me start with the following question, which essentially is addressed to all the speakers. I think the last year was very special, where we really saw how a technology, specifically the mRNA vaccines, have received such focused attention and so many resources that really accelerated the development and turned things upside down. And how do you consider the situation concerning your own technologies and research? Is it really a matter of shared effort and similar to what we've seen last year, that will, with enough time, with enough money and enough people, we can see results and translation very quickly? Or do you think that there's still fundamental issues that require more research and more addressing more fundamental challenges?
Maybe I can comment. I think if I can comment, what I kind of took from the last year is the gap that we have in the regulation in this field. And I think that now that we realize that if we need speed processes, it's possible to do. I think that's one of the hurdles in the nanomedicine area. We have wonderful technologies, but it's very difficult to translate them. And I think there is a very uncertain, I would say, big uncertainty of what are the demands even for translation of nano products. I think this is a gap that we will need to tackle if we want to see more products in the market.
So for me, I think, I'm the closest one to infectious disease research. So I think this was a great example of what happened the last year, and I joined Ofra. I think showing the combination of really strong research and a lot of money invested in one serious problem that needs answer, and combination of a little bit moving or focusing or even boosting a little bit the regulatory aspects to lower the bar a little bit and to bring up front therapies quickly to the market. I think those are things, looking at the questions that I raised in my talk, it's clear that if this will happen, we will have solutions. So we're suffering from both regulatory aspects and funding in terms of fighting microorganisms. And I think the COVID research, I already see it with the interest of students. I mean, microbiology, there's the microbiome that it's a big hype, everybody knows it, but antibiotics, virology wasn't in the focus of many students. And you see that what happened in the last year and a half is really shifting towards this in terms of interest. I hope it will stay, and people will not forget it in a year or two, and because we need to invest in this. It's a challenge that we're all going to face in recent years.
And for our Chinese colleagues, do you see the same processes taking place in China? Do you compare the similar effects happening in the regulatory processes?
You mean for the treatment, or just the imaging?
In both. Do you see any internal discussions about modifying regulatory, expediting processes, modifying regulatory processes, shifting in attention in students, in companies?
Yeah, and now in China side also want to just shift and modify some regulations and the experiment procedures according to the process proceedings of the nanomedicine method.
Thank you. Itamar, unmute yourself.
Yeah, in fact, I have questions to Ofra as well as to Ehud Banin. Regarding Ofra, I'm quite impressed by this. Let me, just a minute. I want to reduce the volume. Okay. I think that there's elasticity differences between normal cells and cancer cells are not very serious. It was twofold, threefold. Is it really a parameter, except for one kind of cells? Is it really something that one can rely about the significance?
I think at least from our experiments, we saw that clearly there are differences. It's very cell type dependent. What I showed you is some examples from the literature, but actually there are some cells that show even greater differences. And I think if we find the right window of particles, then you can kind of increase these differences. So in terms of uptake, you can actually see quite big differences. And as large, if you get larger with the size of the particles, it's actually more driven by the formability. For smaller particles, it's actually a combination of other mechanisms. Sometimes it's adhesion and endocytosis. So at least for submicron, we see that it's enough to provide selectivity. Again, ex vivo. In vivo,
Yeah,
we still don't know.
I think that one problem that we don't have the bridge between, for sure, that particle size, particle shape, particle nature will affect the permeation. But I would like, and as a chemist, I think that to modify one kind of particle with some entities that change the world of permeation would be a very important path to follow. And this link is missing.
I didn't show some of the results. We actually have a paper discussing two types of particles. One is more elastic than others, soft and stiff, with the same composition, that we show that only by tuning the particle elasticity, we actually modify the penetration into cells. So we have it in the other way around too. We quite confident that this is biomechanical driven effect. I agree that we still don't know how valid it would be in vivo, where we have such a complicated biological environment. But again, at least from our experiments, it's very clear that we have this interrelationship between biomechanical and the formability of cells and uptake.
Thank you, thank you. Sure. Now I will go and move to Udi. I think that one problem when we deal with the cytotoxicity or that originates from an effect of a particle on a cell to kill it, we are to some extent hand-waving, and this is based on, I would say, on my own experience, that we don't invest too much in identifying the reactive species that participate in the degradation of the cells. I think that what we are doing is to some extent, we are looking for hydroxyl radical, peroxyl radical, and we move about how they kill the cell or speculate how they kill the cell. But I think that we miss a lot on the way how ingredients in the cell or in the chemistry around participate in the formation of this reactive species. Am I right?
You're completely right, completely right. I agree with you. There's a lot of hand waving on the activity, the antimicrobial activity of nanoparticles. We use reactive oxygen species as a big word, that this is the activity, but as Ofra mentioned, I think there's also mechanical interactions that damage. And I think this is a major question. I can tell you that we're trying to attack this from using the bacteria as a tool. We are actually trying to look at resistance mechanisms against nanoparticles. So like what I showed you for antibiotic, I'm not hoping that it will happen, but eventually it will happen that upon sub-inhibitory concentrations, 2 nanoparticles will start seeing resistant mechanisms. And I hope that through these resistant mechanisms, we might want to start getting information how the cell is responding and what cellular processes may be challenged using these nanoparticles. And hopefully this can maybe start giving some answers what's activated or disactivated within the cell. So I hope that we'll have some more intelligent answers, but I completely agree with you. It's really a black box, and we're trying to look at the effect of different structures. This is actually something I started to do now with Uri, with his nanorods, compared to the spherical nanoparticles of the metal oxides that we have at hand. So there's many questions on both mechanical and the exact species outside and inside the cell that might be triggered. But this is, it's a lot. Currently, I'm sorry to say, there's a lot of unknowns there.
Thank you. Thank you.
Okay. Anybody from the audience? Last opportunity. Okay. If not, then I want to conclude this session by first thanking again all the participants, and mention again that I think one of the main goals behind these conferences is really to build long-lasting collaborations. And so now that this entire session and the rest of the sessions were actually recorded, I think it's a good opportunity to make these talks available to students. I think all the Israeli universities are very active in the last several years in establishing strong collaborations with Chinese universities and institutes. And so I think it's very a big motivation to further these discussions into actual joint projects and ongoing discussions. So I hope this session contributed just a bit to further these long-lasting ties. So it was a pleasure for me. And Professor Wu, do you want to say a few words?
Okay, thank you. Also, it's a great honor to just chair these afternoons and in China side, and announce the top 4 speakers. A very nice and interesting talk. And this, I think, for both two letters sides, Israel and China, learn a lot about the nanobiology and the nanomedicine's new progress. And hope in future we have more collaboration with each other. And then, if possible, after the pandemic COVID-19 deceased, maybe we can just have the person by person, just talk about the scientific topics each other. So thank you all presenters 4 presentators and also the chairmen and the moderators, and especially Professor Itama Wiener. So let's just welcome to Itama Wiener to get our closing the remarks. Okay.
Okay. Thank you, Professor Wei. I think that I'm on the way to bring these 4 webinars to the end. But I think that we are just at the beginning of trying and to continue our outstanding collaboration with China. And these 4 webinars, under the circumstances that we led them, were overall a success. And I think that everybody learned the strengths and the possible contribution of nanotechnology to science. I really think that these webinars should be should spark our continuous relations with China. And I look forward to the future where the results of this corona pandemic will terminate and we will be able to meet one another, to have mutual meetings, and to have real face-to-face collaborations and interactions. While finishing this meeting, I think that I should thank again, first of all the chairpersons, moderators of all 4 webinars. Again, I would like to thank the staff of the Israel Academy and of the Chinese Academy, who helped us a lot in raising these efforts. I know how complicated it is, and I really, really, really would like to thank everybody for the help that was given to us to succeed and have these conferences. So thank you very much.
Okay, thank you. See you.
Thank you.
Thanks, everyone.
Bye-bye, everybody.
Bye-bye.
Bye.
Bye-bye.